So good morning students, myself Dr. Rajat Singh, and today we are going to cover the second part of neural control and coordination. So the topic we are going to cover in today's lecture, yesterday in the previous lecture, actually we didn't complete the last topic, which was like a generation and conduction of impulse. So that was the topic which was remaining from the previous lecture. So today we are going to complete this topic first, and after that the next topic we are going to cover is the transmission of impulse. And the last topic we are going to cover of this chapter is the central nervous system. Central nervous system. So these three topics we are going to cover in today's lecture. So let's get dive into it. And today we are going to try to depict. Actually, the thing is, in the part 1 of this lecture, every topic was quite a bit of easy, but now this is the conceptual based topics which has come under the part 2. So let's write down the name of the chapter, that is neural control and coordination. So that is the name of the chapter, and first topic we are going to cover is the generation and conduction of nerve impulse. So the thing is, so just write it down: generation and conduction of impulse. So what this topic is talking about, so let's just understand. So the thing is, actually, whenever we what is stimuli is a certain change in environment. So whenever if I talk about the environment, certain change in environment, let's just somebody touch you, from your hand touches you. So what the chemical process is happening in your body? The thing is, whenever somebody touches you, the electrical impulses are generated from the receptors of this skin, and that is being conducted through your CNS, that is your brain, that is your central nervous system. So the thing is, so neural generation and conduction are both very important. Both are very important process which need to be understand very well. So the thing is, generation of impulse should be needed because by this phenomena you are just sensing each and every stimuli, the changes in different environment, whether it is sight seeing or hearing or touching or thermal regulation or pressure receptors, every kind of receptor you have in your body, and that are need to be stimulated by some stimuli. So that stimuli is stimulating your receptors, and that receptors are conveying that, generating that impulse and just conveying that impulse to your CNS. So the thing is, how impulse is generated. So first, let's just take you people already know this thing about that the neurons are responsible, the organized network of neurons are responsible for neural system. So the impulse is generated by receptor, and the impulses is carried and conveyed to the CNS by neuron. You know this thing already, that we have already discussed in our previous lecture. So the thing is, when I draw the part of neuron, that is our axon. So this is the axon and this is the first part. So you can write it the point first. So the thing is, this is the axon part, and the axon part is filled with a fluid known as exoplasm. That is similar to cytoplasm, and this is the membrane of axon. And the thing is, firstly, whenever axon is at resting phase, there is a charge across the membrane like this. Positive charge is outside the axon membrane like this, as you can see, like this, and inside the axon membrane there is a negative charge on both the membrane. Actually, axon is a cylindrical structure, but as you can see it's just parallel lines. So there is a charge, like inside there is negative charge, as you can see, and now outside the membrane there is a positive charge. You can also see like this. So the thing is, there are some kind of protein, membranous protein, which are present on the exon membrane as well. So let me just write it down like this. So what are those proteins? So first one protein is like this. I am drawing it with red color. So that red protein channel is known as sodium potassium pump. So that red protein is sodium potassium pump, which you already know. This is a actually this does active transport which requires ATP. So the next channel that is present on exon membrane is, I am just drawing it with black color, and the next one is I am also drawing it with black color. So this one is sodium, yani that's why I am writing Na, sodium VGC, and this another black one is potassium. I am writing denoting it with K, and VGC. So what do you mean by VGC? VGC stands for voltage gated channel. So this is the first condition in which you can see the exon. This you can see exon is the potential. Actually, there is a charge on outside the surface on the exon membrane, and inside the exon membrane you can see the negative charge is over here. So this is the resting phase. Stimulus has not been generated, and if stimulus is absent, then there is no action potential. So let's just understand what is the use of these pump and channels are present over here. So the thing is, you can see the exon membrane is charged by negative inside and positive outside. So at resting condition, I call it resting condition. So at resting condition, this negative charge inside and positive charge outside is maintained by a sodium potassium pump, which are always open. So here I am writing with point. So at resting position, resting stage, at resting stage, negative charge on inside and positive charge on outside. As you can see, this is the resting stage. Next point that comes under this is sodium potassium channel are always open. That means these pump is responsible for negative charge inside and positive charge outside. Such condition is known as polarized state. So that condition is known as polarized state, when positive charge is outside and negative charge is inside. So that condition is polarized state. And sodium potassium channel I am writing here is sodium potassium pump are always open. So sodium potassium channel are always open, and they are just maintaining this state known as polarized state, and maintaining polarized state of exon. So you can see polarized state, this phase is known as polarized state. So what the sodium potassium pump is doing, actually sodium potassium pump, you can see in exoplasm there are some sodium ions and many are potassium ions. So actually what this pump is doing is, it is influxing potassium ion and outfluxing. That means it is sending sodium outside, sodium ion. So how this charge is maintained? Because both ions are positive. Actually the thing is, the potassium ions are 2 in number and sodium ion are 3 in number. So you can see 3 sodium are outfluxated by a sodium potassium pump. So 3 positive charge are getting outside and 2 positive charge are getting inside. So you can see less positive is inside and more positive is outside. So that is responsible for negative charge inside. And this exon membrane is impermeable for sodium ion. So that exon membrane is impermeable for sodium ion. So here I am writing it. So the point is exon membrane is impermeable for sodium ion and permeable for potassium ion. And again, in exoplasm there are negatively charged protein are present which are also responsible for such polarized state. So here I am writing is negatively charged proteins are also present in exoplasm, and the membrane of exoplasm is also actually impermeable for the negatively charged protein, and membrane of exoplasm is impermeable for such proteins. So these kind of pointers you need to remember at resting stage, and the thing also is at resting stage you can see the positive charge outside and negative charge inside. So such kind of charges are developing a potential. So that potential is known as resting potential because the exon is at resting state. So here I am writing with another color. What is resting potential? Resting potential. So resting potential is potential that is maintained across the membrane of exon. So potential that is maintained across the membrane. So the thing is, across the membrane there is a potential, so which is known as resting potential because this exon is at polarized state. So this is the first stage. So you have understood. So now what's going to happen is, there is a generation of action potential because the stimulus has come in contact with dendrons, and the dendron is generating somehow the action potential, and now this condition is known as polarized state. And this condition is known as polarized state.
चार्जर है क्या आपके पास? मैं
कौन सा चार्जर चाहिए उसको?
BSC
सी है मेरे बैग की आगे की चेन में से निकाल लेना।
उन्होंने मुझे कहा था कि अभी ले लो, इसीलिए मैंने आपको डिस्टर्ब किया।
नहीं, मैं तो काट के जोड़ लूंगा, कोई दिक्कत वाली बात नहीं। इसी तरह मेरा बैग है ना, उसमें आगे छोटी चेन होगी, उससे जस्ट सेकंड वाली चेन में देख लेना चार्जर पड़ा। Sorry for the disturbance. Actually, so sodium potassium pump is working like this, and there are also negatively charged proteins are present. Let me try it again. So the sodium potassium pump are just outfluxing sodium 3 ions and 2 ions of potassium are influxed in the exoplasm, and also negatively charged proteins. So this whole concept is maintaining the polarized state. So when stimulus is stimulating the receptor, that is also generating action potential. So I hope you have written all this. So now I'm going to be explaining how action potential is being generated. So now action potential is being generated like this. So this is the exoplasm, as you can see. So now this channel are being open forever and always open. So now what's going to happen is, when the action potential is generated, this voltage gated channel, sodium voltage channel are influxing lot of sodium ion inside the exoplasm, which is responsible for generating action potential, and action potential which is also generating the positive charge inside the exoplasm, let me just draw the diagram. So here is our sodium VGC. So VGC is influxing sodium ions a lot. So these channels are open. So these channels are open by fluctuation in the voltage. So actually you are saying by fluctuation of charge or voltage, you can say that is why these channels are not voltage gated. So they are very sensitive to fluctuation in voltage. So that is why these are open now, and they are just influxing sodium ion a lot inside this. So sodium are coming inside this. So the thing is, now let me just draw positive. So now this is your exon, and now sodium voltage gated channel are influxing sodium ion a lot inside, and again there are also another, this is potassium VGC channel which are outfluxing actually potassium. So they are outfluxing potassium, so like this potassium ion inside. So now you can understand the condition. 3 sodium ion are coming inside, and 2 potassium are going outside. So these voltage gated channels are trying to generate the positive charge inside, because 3 sodium are coming inside and 2 potassium are going outside. So these are trying to generate positive charge in the exoplasm across the membrane. So like this. So this is going to happen. So after some time, you can see, now this is your exoplasm. You can see at rest of the remaining membrane of exon, you can see positive charge, but actually now here you can see the negative charge, and inside this you are generating positive charge because sodium ions are continuously influxed by sodium VGC, and potassium are outfluxed by potassium VGC. So you can see like this. So you can see now at this point you can see inside the membrane there is a positive charge on both sides and negative charge on outside the membrane. This is due to the sodium voltage gated channel and potassium voltage gated channel. So now they are generating positive charge inside the exoplasm. So now action potential is being generated. Actually at rest condition you can see at rest condition such potential is known as resting potential, but now action potential has been generated. So now this is second one and this is third one, and here I am writing action potential is generated. So you can see at resting position that potential is known as resting potential, but now the action potential, this potential across the membrane now is action potential, and this condition is known as depolarized condition of exon. So here I am writing this condition is known as depolarized condition. So this condition is known as depolarized condition. Actually in rest of the membrane this is polarized condition, but in this part I am talking about this is depolarized condition. So now you have understood how positive charge is coming inside and outside it is getting negatively charged. So again, so now the action potential is moved this much, and now this part has to be covered because that is the conduction part of action potential. So now sodium are getting inside, and again at this part of exon you can see there are also sodium and potassium voltage gated channel are also presenting, which are doing such same thing. Sodium VGC are just influxing sodium 3 and the outfluxing potassium ions. So same thing is going to happen over here, and this is the polarized part and this is the depolarized part. So this action potential is now being conducted to the next part. So what's going to happen here is, as you can see, the fourth part. As you can see the fourth part, here I am. Plus, 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 plus. Plus, 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 plus. Plus, 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 plus. Plus, 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 plus. So here you can see, now I am talking about exon. So this part has been, again, the negative charge is inside and the positive one is inside. So that is maintained by, again, the sodium potassium pump, which I have already talked about earlier. So this condition, after being depolarized, now it is being repolarized due to continuously active action of sodium potassium pump. So this condition is known as repolarization of exon. So this part is repolarization of exon. Let me mention it. Repolarization. And this action potential now is being conducted to this due to sodium VGC channel, and now this is being conducted further. So same thing is going to happen like this I have mentioned before. So this part is repolarization and this is depolarization. Depolarization, and this is again polarized. So now you have understood the thing that how action potential is generated due to stimulus. Stimulus is stimulating, and now they are collecting from the receptor, and now from the exon part the action potential is being generated, and now it is being conducted in the form of impulse. So as you can see, this is the fourth part. So in fourth part I have completed all this thing. So I hope you understand this topic. Generation of conduction of electrical impulses is like this. So let me come over to the next topic, that is transmission of impulse. So that is also a very important topic you need to understand. Actually, this is generation and conduction of nerve impulse, but you don't understand. The conduction is being the conduction of impulse is being carried by multiple neurons. So there should be a gap between some neurons. So that a part we are going to talk about in transmission of impulse in the next. In this topic, you have understood the generation of impulse. Let me draw. So the thing is, you already know that this is the axon, okay? Like this, and this is synaptic knob are present over here. So the conduction is being of electrical impulses are carried in axon part like this. This is myelin sheath. So the conduction is being carried in the axon part. So what happens when the impulse come at synaptic knob? Synaptic knob is a bulb-like structure which is present at the termination of branch of axon. So synaptic knobs are these vessel-like structure. So let me explain. So this gonna be studied in nerve impulse. So here I am making the diagram of synaptic knob. So what's going to happen is the action potential is being come from this direction to this direction towards the synaptic knob. So here I am writing, so action potential is generated and comes is coming towards synaptic knob, which is a bulb-like structure, as we have studied in the NCERT and in the previous lecture as well. So the thing is, they actually synaptic knob is present where the fluid-filled vesicles are present in the synaptic knob, which are coming towards this membrane. So these vesicles are filled with a special chemical known as neurotransmitter. So neurotransmitter is responsible for transmission of impulse. So here you can understand like this. So actually like this. So actually first thing you need to understand, the transmission of impulse is in two forms actually. So here I am just rubbing it and going to explain the difference between these two. So first, synapses are of two types. So here I am writing is synapse. Synapse. It is junction present between pre-axonal and post-axon, and the gap between them is known as synaptic cleft, and the gap between them what? Pre-axonal and post-axonal part. So between them is known as synaptic cleft. So synaptic cleft. So this gap is I am talking about is it is the synaptic cleft. So here I am writing synaptic cleft. And these vesicles are filled with neurotransmitter, called chemical known as neurotransmitter, and this is the axon part and this is the bulb-like structure synaptic net. So what is the synapses? So synapses is the junction. So this is the point junction. As you can see, a junction present between pre-axon like this and post-axon like this. So post-axon is the next one, and which is going to collect the neurotransmitter from this and going to generate action potential again. And so this is the gap between the two neurons. So this gap is then called as between them. That them means what? Pre-axonal part and post-axonal part. So this gap between them is known as synaptic cleft. And these 3 points, pre-axonal and post-axonal plus synaptic cleft, is known as synapses. Now synapses are of two types. The first one is the chemical synapses and the second one is the electrical synapses. So here I am writing and the difference as well. So what is the difference between them? After that we are going to understand what is happening. So the synapses are of two types. First one is chemical synapses. Second one is electrical synapses. So what is the difference between these two? Let me explain. The chemical synapses, chemical is involved, and like this I have shown in the diagram. So chemical is involved, that is known as neurotransmitter. So neurotransmitter are involved. Electrical impulses are involved, that means there is no neurotransmitter. So you can write like this: electrical impulse is involved. Okay. Now the second point is, this chemical synapses is comparatively slow. Actually it is fast, but if I compare with electrical synapses, it is comparatively slow. So here I am writing this is comparatively slower than electrical. this is faster than the chemical one. So this is the difference, and the gap between the synaptic cleft, the actually the post-axonal and pre-axonal part are in the close proximity. Proximity means I mean to say the distance. So pre-axonal and post-axonal gap is very less. Third one is pre and post axonal gap, that is synaptic cleft, is more. Pre axonal and post axonal gap is more. Comparative in comparison to electrical synapses, the gap between this is more. So here I have written the difference between electrical synapses and the chemical synapses. So let's come over this topic again. So this is the example of chemical synapses. The diagram is already being shown. So now what is going to happen? These vesicles are filled with vesicles are filled with neurotransmitter. So these vesicles are filled with neurotransmitter. So what's happening is action potential has come to this point, and now these are generating some vesicles. That vesicles are filled with fluid known as neurotransmitter, and now the vesicles are moving toward the membrane, the pre-axonal membrane, and these vesicles are getting fused. So these vesicles are getting fused with synaptic nerve membrane. So vesicles are getting fused with membrane of synaptic nerve. So synaptic nerve, these membranes getting filled, these getting fused, and with fusion, the neurotransmitter are coming outside, and these receptors are here for reception of neurotransmitter. These receptors are now being stimulated by neurotransmitter. So here you can see in this cleft the neurotransmitter is getting filled like dot 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 dot. I have drawn over here, and these receptors are now the post-axonal part is now the part of the next neuron, and these next after reception they are going to generate again action potential, which is being generated and conducted through the next axon. Excuse me. So what's happening with this marker actually? So here I am again. So these receptors, here I am writing with another color. These receptors are now stimulated by neurotransmitter, and in the next axon part, these neurotransmitter is now sensitizing these receptors present on the post-axonal part, that I mean to say the next neuron, and which are going to generate the action potential again in the next neuron, and in this pattern the transmission of nerve impulse is being carried in our body. So this is the example of chemical transmission, and I have shown the difference between what is chemical and what is the electrical impulses. So it is a little bit faster than the chemical. Well, actually they are both. If I talk about the electrical synapses, the transmission is like this. There is no involvement of neurotransmitter in the synaptic nerve. All is going to happen is just action potential actually the distance between the gap of synaptic cleft is very less in electrical synapses, and the action potential is being carried straight, and it is going continuously from synaptic nerve to the next neuron. So in that way, electrical synapses and electrical transmission is very faster than the chemical one, because there is no involvement of neurotransmitter. So let's just come over to the next topic, and that is our central nervous system. So central nervous system. So I am going to draw down a schematic diagram of our central nervous system, and I will dictate everything. All you need to do is just write down, because in today's class we have I am out of time. So here I am drawing like this. Let me just label it down. So this is this is our brain, and I am going to discuss here in central nervous system. I am going to discuss the various parts of our brain. So this is actually, you can see these are ridges, and some are present. So this is our brain, and I am going to discuss here the parts of the brain and which part is going to perform which function. So first of the thing is, actually I need to draw another one, is like this. So as you can see from above, you can see the brain is in the half part, but this is the sideways view of brain. So you can see in this brain, so let me draw just gyrus SI over here. These are the ridges, and some kind of, actually the lines are present on the cerebrum. So this part is known as cerebrum. Cerebrum is in two parts. If you view brain from upside, then it is divided into two parts, and this is actually right cerebral hemisphere and the left cerebral hemisphere. The right one is right cerebral hemisphere, and this is the left one, left cerebral hemisphere, and this is our spinal cord. So actually the thing is, this cerebrum is divided into two parts, the left one and the right one. So now this cerebrum has multiple areas over which actually actually the every impulse, whether it is from touch or temperature regulation and everything, hearing, sensation, which is come towards the CNS. So now what I am talking about is every area is present over here. So this is the optic lobe, this is the actually there is a present over olfactory receptor and a receptor for our hearing, receptor for our talking, receptor for our Wernicke's area over here, which is responsible for our vocabulary generation from our tongue. So let me come over to the next thing. This part is known as corpus callosum. So this part is known as corpus callosum, and this part is known as your hypothalamus, and this part is known as your thalamus. This part is known as your midbrain, and this cerebrum, corpus callosum, thalamus and hypothalamus are part of your forebrain. And now let's talk about hindbrain. Actually, this part is known as your pons. This part is known as your medulla oblongata. So medulla oblongata, and this part is known as your cerebellum. So let's talk about each and every part. Because we are out of time, so we have to cover this topic. So all you have to do is just write it down what I am talking about. So the cerebrum is also known as the gray matter. It is made up of gray matter. So why it is gray? Because myelinated neurons are present which are responsible for gray matter. And this part is known as cerebral cortex, and inside part is known as cerebral medulla. All I am trying to explain is this layer of brain outside, which is outside, is known as cerebral cortex, and this inside part of the brain is known as cerebral medulla. For our basic understanding, I am just telling you. So this cerebral cortex, cerebrum, inside cerebral cortex, there are myelinated nerve fiber which are myelinated. So that is why this is the gray matter, or this is made up of the gray matter, because myelinated neurons are present. Inside the cerebrum, there are sensory and motor neurons are also present. Motor neurons and sensory neurons. So every kind of sensation I have already talked about. The touch receptor, the pressure, temperature, each and everything. Each and every impulse is being generated by neurons are carried toward this part, because there are many parts are responsible for analyzing. Actually, brain is also known as the command and control system of our body. So it is so every impulse is coming towards the brain. So Wernicke's area is present, olfactory area is also present, and each and every area is also present, which is responsible for some analysis related to according to that stimuli. So again, the next part is corpus callosum. As you can see, the brain, the cerebral, is divided into two parts like this. So right cerebral hemisphere and the left one. So the corpus callosum is responsible. Actually, it is a present layer in the form of layer which is made up of fibers or nerve fibers. So like this, these two hands are like left and right cerebral hemisphere, and inside this you can see. Between them you can see there is a gap, and this gap is being covered by a layer of fibers which is known as corpus callosum, which is responsible for coordination and synchronization between the left and the right cerebral hemisphere. So this corpus callosum is responsible for this. So the next part comes is like thalamus and hypothalamus. So if I talk about this roof part is covered inside the cerebral hemisphere. This roof part is covered by corpus callosum, and this thalamus part, as you can see, if this is a roof, and this roof is made up by corpus callosum, and these walls are going to be made by thalamus. So now you can understand what is thalamus. Thalamus are, if this is the midbrain or forebrain combination roof. So you can see these walls are thalamus, and the corpus callosum is the roof of this room, and the hypothalamus is the floor of this room. So in that way you can understand what is this. So thalamus is this. Now let's come towards hypothalamus. So hypothalamus actually So hypothalamus, if I talk about hypothalamus, you already know hypothalamus secretes some various hormones, hormones, which is actually pituitary gland is in the direct control of hypothalamus. So hypothalamus is present over here. So it has some ganglion. Ganglion is the accumulation of some neurons, cytons, and they are generating some hormones. And they are generating hormones, and they are controlling the entire, not entire pituitary gland, but entire pituitary. But you can say they are indirectly or indirectly controlling pituitary gland. So that will come under hormone coordination and integration chapter. So hypothalamus. So here we can understand hypothalamus is regulating the eating and drinking urges and temperature regulation. So here I am writing. So hypothalamus is controlling eating and drinking urges, controlling eating and drinking urges and temperature regulation So now you have understood. So let's come over the midbrain part. So midbrain part, you can see midbrain part is over here. So it is somewhere between the thalamus and hypothalamus and between hindbrain. So this part is covered under hindbrain. Hindbrain is made up of pons, medulla oblongata and cerebellum. So these three parts are pons, medulla oblongata and cerebellum are coming under hindbrain. So midbrain is a very small portion which is present over here. So midbrain is like over here. So it is between some thalamus, hypothalamus and hindbrain. You can say hindbrain. So this midbrain. So where various nerve and sensory and motor nerves are present. So actually there are four we can say lobes are present, which is in our NCERT has explained, which are known as corpora quadrigemia. Here I am writing, it has four lobes known as corpora quadrigemia. So four lobes are present in the midbrain, which is corpora quadrigemia. And now let me come over the hindbrain part. So the hindbrain part, as you can see, this swelling, this bulging is known as pons, and this further continuation of this spinal cord is known as medulla oblongata. So and this part is cerebellum. Cerebellum is responsible for balancing of body. So here I am writing responsible for body balance. Okay. And this duct is known as cerebral aqueduct. Here I am writing with it some other color, that is cerebral aqueduct. So what is the meaning of cerebral aqueduct? Actually it is the duct which is carrying the CSF. Carrying the CSF, that is cerebrospinal fluid, is being carried inside the cerebral aqueduct. Actually the brain is flowing under a fluid, inside a fluid, so which is responsible for shock absorption and just actually which is providing a bio and force. So that fluid is known as cerebral fluid. So cerebral fluid, so actually it is providing bio and force, and it is just protecting it from sudden damage and sudden if we do like this. So brain is not moving like this because inside brain cerebrospinal fluid is present outside the brain, which is just controlling the movement of sudden movement of brain. So the cerebral aqueduct, inside cerebral aqueduct, CSF is moving. So here I am writing CSF is flowing. Again, so let's come over the pons and the medulla oblongata. So pons and medulla oblongata has various centers, and in medulla oblongata in NCERT it is being taught in NCERT that it has respiratory and cardiovascular centers over medulla oblongata, which are responsible for controlling our cardiovascular activity, whether it is sympathetic or whether it is going to be parasympathetic. That all is come from medulla oblongata over here. And if I talk about breathing, the respiration center, so respiratory neurotoxic center is also present in the medulla oblongata, which is responsible for deep breathing or quick breathing, fast breathing. All is being controlled by this central nervous system. This is the command and control center of our body. So this is the end of this topic. Actually, this is. I hope that you have understood everything that is being. I have taught everything that is being taught under NCERT, 11th NCERT. So this chapter has come to an end. So this is like this. I could have explained each and every topic in detail. I can help you making notes, but actually I am out of time, and this class is being finished after 5 minutes. So in short and layman language, I have explained all the labeling and all the functions of each labeling. So if you have any kind of doubt regarding this topic, you can ask me anytime. So thank you for the lecture, and this topic has been now completed. So thank you.